Hello everyone, in this video we will learn more about recombinant DNA technology. We will focus on the methods in gene cloning. First, we will look into a few diagrams to understand the methods in gene cloning, and we will describe all of the steps in gene cloning by using plasmid as a vector. Let's take a look into a few diagrams. It consists of isolation, cleave, insertion, transformation and amplification followed by screening. Different diagram, but it will show the same step. Isolation of gene, cleave or cut, insertion, transformation and amplification, and screening. For the methods in gene cloning, we can use the acronym ICITAS to make it easier for us to memorize the step in gene cloning. The first step in gene cloning is isolation of gene. In isolation of gene, we will isolate the plasmid that will serve as a cloning vector from the bacteria E. coli. The plasmid will carry two useful genes, ampR and lacz. The plasmid is a small circular DNA extracted from E. coli, contain two useful genes, the ampR that will code for the ampicillin resistant gene and lacz gene that will code for enzyme beta-galactosidase. Besides, the lacz gene contain restriction site that will be the part of the multiple cloning site. Target gene, or also known as gene of interest, that code for desired protein isolated from human, animal, or plant cell. Example of target gene such as gene that code for insulin from human, gene that code for bovine somatotropin from cow, and also gene that code for beta-carotene from corn. The following step is cleave or cut. Both target gene and plasmid are spliced or cut using the same restriction enzyme. The reason for this is to produce complementary or compatible sticky ends or blunt ends between target gene and plasmid. For example, the enzyme EcoR1 is used to recognize and cut the restriction site of target gene and plasmid to produce complementary sticky ends. Restriction enzyme EcoR1 will recognize the palindromic sequence 5' GAA-TTC 3' of target gene and plasmid. Step number 3 is insertion, in which the target gene or gene of interest is inserted into the plasmid and then sealed by DNA ligase to form recombinant DNA or recombinant plasmid. To complete the insertion step, DNA ligase will seal the phosphodiester bond of target gene and plasmid to form recombinant plasmid. Other than recombinant plasmid, the insertion process also will allow formation of non-recombinant plasmid. During insertion, some of the plasmid will form recombinant plasmid without target gene, and some will form recombinant plasmid with target gene, while the others will form non-recombinant plasmid. The next step is transformation and amplification. Transformation is the process of uptake of recombinant plasmid by bacterial host cell, while for amplification is the cloning process to produce multiple identical copies of recombinant plasmid. The final step in gene cloning is screening. The purpose of this step is to detect the host cell that contain recombinant DNA. The medium culture will contain antibiotic ampicillin and sugar X gel. On the medium culture of blue-white screening, bacteria with recombinant plasmid will form white colonies. Bacteria with non-recombinant plasmid will form blue colonies, while bacteria without plasmid will not survive. The bacteria without plasmid will not survive because it has no ampicillin-resistant gene. Blue colonies form because the bacteria with non-recombinant plasmid have complete lacz gene. This gene that codes for enzyme beta-galactosidase capable of hydrolyze X gel to form blue colonies. While for the white colonies, it consists of bacteria with recombinant plasmid, in which the lacz gene on the plasmid is replaced by target gene. As a result, no enzyme beta-galactosidase is produced and the X gel is not hydrolyzed. The bacteria from the white colonies then purified and cultured for further use.